Our skin and mucous membranes normally provide a barrier against invasion of foreign agents. If damage to our skin occurs, local defense mechanisms mount the initial attack on invaders. If deeper penetration of pathogens occurs, the adaptive immune system relies on lymphocytes to provide a two-pronged attack on invaders and infected body cells. One response, cell-mediated immunity, occurs when T lymphocytes directly attack and destroy diseased or foreign cells. The other process, antibody-mediated immunity, does not involve direct lymphocyte destruction of invaders. Instead, B lymphocytes release antibodies into the blood and interstitial fluid to neutralize pathogens. T lymphocytes mature in the thymus to become either cytotoxic T cells, which directly attacks diseased or foreign cells, or helper T cells. Helper T cells aid the immune responses of both cell-mediated and antibody-mediated immunity. B lymphocytes mature in the bone marrow to become plasma cells which produce antibodies. During the maturation process, these lymphocytes develop the ability to carry out adaptive immune responses through the insertion of distinctive proteins, called antigen receptors, into their plasma membranes. B lymphocytes, when activated by the secretions of helper T cells, divide by a process called clonal selection into antibody-producing plasma cells and B memory cells. Cytotoxic T cells are similarly able to clone themselves into active cells and memory cells. Helper T cells have a central role in the immune response by releasing stimulatory molecules known as interleukins. These interleukins greatly increase the cloning of plasma cells to stimulate a high production of specific antibodies and cytotoxic T cells to attack invaded cells. The number of memory cells also increase substantially. Most active cells eventually die after the immune response has been completed. Memory cells remain inactive in case the same pathogen enters the body at a later time. This enables them to rapidly respond to the next invasion. Pathogens such as bacterial cells or even cellular components that are recognized as foreign provoke immune responses and are called antigens. Small parts of a large antigen molecule, called epitopes, act as the triggers for immune responses. Each epitope on an antigen can activate a specific T cell or induce production of a specific antibody from a B cell to target different antigens on a single pathogen. An amazing feature of the antigen receptors on both B and T cells is its ability to recognize and bind to at least 1 billion different epitopes. The T and B cells are able to differentiate between healthy and diseased body cells or foreign cells because all body cells, except for erythrocytes, produce a membrane marker known as the Major Histocompatibility Complex 1, or MHC1. When a body cell becomes infected by a pathogen, the pathogen's antigen is added to the MHC1 marker, creating a signal that targets the infected cell for destruction by immune cells. If the pathogen has not invaded a body cell, defensive cells, including B lymphocytes, phagocytes, and dendritic cells, known as antigen-presenting cells, or APCs, consume and digest the invader. Fragments of the pathogenic antigen are attached to a second version of the MHC identifier, MHC2, which is inserted in the APC's cell membrane. These cells expose the antigen in a way that activates helper T cells, which in turn promote a higher level of adaptive immune response. When a cytotoxic T cell is activated by the actions of APC and helper T cells, it recognizes the MHC1 complexes on the infected body cell's surface. Cytotoxic T cells then release two molecules, perforin and granzyme, in order to destroy the infected body cells and the pathogens that infect them. Perforin creates pores in the target cell membrane. The pores allow extracellular fluid to enter the cell, causing it to swell and potentially rupture the cell membrane. Granzyme then enters through the pores and causes the degradation of its proteins to ensure cell death. Cloned B cells do not attack individual cells. Instead, after they become plasma cells, they greatly increase their production of antibodies. These antibodies are released into the plasma and extracellular fluid, where they will match and bind to the target antigen. Antibodies are Y-shaped structures with unique antigen-binding sites on the tips of two of the arms. This allows an antibody to perform different functions depending on the type of binding site it possesses. They may essentially prevent invaders from interacting with normal body cells, or mark foreign cells for destruction, and activate our defensive cells into specific action. The two-pronged approach of our adaptive immune system, either cell-mediated immunity through the action of cytotoxic T cells or antibody-mediated immunity initiated by B cells, provides us with an amazing defense arsenal against disease-invading pathogens.